Welcome to my lab one, my human physiology lab one study guide. Today we'll be identifying tissues in the laboratory, review cell structure, including functions, organelles, review concepts of tonicity, and understand how it's affected by osmosis. Review experimental protocols. All right, we're gonna start with number one. And number one. Cell membrane protection, interaction, transport. Cell membrane protection, interaction, transport. Cell membrane protection, interaction, transport. Mitochondria, ATP. Mitochondria, energy, ATP. Mitochondria, energy, ATP. Mitochondria, energy, ATP. Golgi apparatus, post office, package, send. Golgi apparatus, post office. Golgi apparatus, post office. Golgi apparatus, post office. Endoplasmic reticulum, metabolism, and protein lipid transport. Endoplasmic reticulum, metabolism, protein, and lipid transport. Endoplasmic reticulum, metabolism, protein, and lipid transport. Ribosomes, protein synthesis. Ribosomes, protein synthesis. Ribosomes, protein synthesis. Ribosomes, protein synthesis. Lysomes and peroxisomes, digestion and sanitation. Lysomes, peroxisomes, sanitation. Lysome, peroxisomes, digestion / sanitation. Cytoplasm matrix chemical reactions. Cytoplasm matrix chemical reactions. Cytoplasm matrix chemical reaction. Cytoplasm is mostly thick water to hold organelles, gives place for chemical reactions. Cytoplasm mostly thick enough to hold organelles, gives place for chemical reactions to occur. Nucleus house genetic material. Nucleus houses genetic material. Nucleus houses genetic material. Nucleus houses genetic material. Nucleolus houses RNA. Nucleolus houses RNA. Nucleolus houses RNA. Vacuoles for storage. Vacuoles are for storage. Vacuoles are for storage. Ribosomes protein. Ribosomes protein. Ribosomes protein. Ribosomes protein. Ribosomes protein. Next we got simple squamous epithelial tissue. And then we got simple cuboidal epithelial tissue and simple columnar epithelial tissue. And we got neuron, skeletal muscle, cardiac muscle, smooth muscle, adipose tissue, dense regular connective tissue, bone, blood, and hyaline cartilage. This is what simple squamous epithelial tissue looks like. Simple squamous epithelial tissue. Simple squamous epithelial tissue. Simple squamous epithelial tissue. Simple cuboidal 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 epithelial tissue. Simple columnar 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 epithelial tissue. This is neural tissue. Neural tissue. Neural tissue. Neural tissue. This is neuron. Neuron. This is skeletal muscle, skeletal muscle, skeletal muscle, skeletal muscle, skeletal muscle, skeletal muscle. That's skeletal muscle. Cardiac muscle, cardiac muscle, cardiac muscle, cardiac muscle, look for the intercalated disc in the cardiac muscle. Cardiac muscle, cardiac muscle. Smooth muscle, smooth muscle, smooth muscle, smooth muscle, smooth muscle, smooth muscle, smooth muscle. Adipose tissue. Adipose tissue can also look oily. Adipose tissue, adipose tissue, adipose tissue, adipose tissue, adipose tissue. It's used for storage. Dense regular connective tissue, dense regular connective tissue, dense regular connective tissue, dense regular connective tissue, dense regular connective tissue. Bone, 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 bone. Blood, 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 blood. Hyaline cartilage, hyaline cartilage, hyaline cartilage, hyaline cartilage, hyaline cartilage, hyaline cartilage, hyaline cartilage. That's it for that section. Next, we'll be going to laboratory 2, diffusion and osmosis. What is most of the human body made up of? It's made up of water. What is most of the human body made up of? Water. What is most of the human body made up of? Water. Where's water found? Intercellular water and extracellular water. There's intercellular water and extracellular water. There's intercellular water and extracellular water. Intercellular water and extracellular water. Extracellular water is matrix, tissue, spaces, and plasma. 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 Also, in extracellular matrix, connective tissue, collagen, elastin, ground substance. Also, in extracellular matrix, connective tissue, collagen, elastin, ground substance. Diffusion of the cell membrane. Cell membrane is composed of non-polar, not charged phospholipids. Anything that is also non-polar oxygen, carbon dioxide, or that is lipid-soluble steroid hormones can pass through. Large molecules of glucose and charged molecules and ions. Sodium, chloride, potassium must use ion channels, proteins that span the cell membrane. Diffusion in the cell membrane. Cell membrane is composed of non-polar, non-charged phospholipids. Cell membrane is composed of non-polar, not charged phospholipids. Anything that is also non-polar oxygen, carbon dioxide, or lipid-soluble steroid hormones can pass through. Anything that is non-polar, that is lipid-soluble, can pass through the cell membrane. Large molecules, glucose, and charged molecules and ions, sodium chloride, potassium, must use ion channels, proteins that span the cell membrane. Rate of diffusion. How big is the concentration gradient? How permeable is the membrane to the molecule diffusing? What is the temperature of the solution? What is the surface area of the membrane where diffusion must take place? If the diffusion concentration gradient is big, then diffusion, there won't be as much of it. If it's permeable, it'll diffuse pretty fast. All right, osmosis. The net diffusion of water across the cell membrane. For this to occur, the membrane must be more permeable to water than it is to solute present. If water is the only thing that can move through the membrane and the goal is to equilibrate the number of solute particles per volume of water, then water will move across the membrane from the solute that is less concentrated to the solution that is more concentrated, until both solutions have the same number of solute particles per volume of water. Osmosis goes from less to more, I think. Osmosis. If the net diffusion across the cell membrane, osmosis, the net diffusion of water across the cell membrane. For this to occur, the membrane must be more permeable than it is to the solute present. If the water is the only thing that can move through the membrane, and the goal is to equilibrate the number of solute particles per volume, then the water will move across the membrane from the solution that is less concentrated to the solution that is more concentrated. Osmosis. Water will move across the membrane from the solution that is less concentrated to the solution that is more concentrated, until both solutions have the same number of solute particles per volume of water. Osmosis. Water moves across membrane from one solution that is less concentrated to a solution that is more concentrated. Osmosis. Water moves from a solution that is less concentrated to a solution that is more concentrated. Osmosis. Water moves from a solution that is less concentrated to a solution that is more concentrated. Next is tonicity. Isotonic. Two solutions have an equal number of particles per volume of water. That's called balanced. Tonicity. Isotonic, hypertonic, and hypotonic. Tonicity. Isotonic, hypertonic, and hypotonic. Tonicity. Isotonic, hypertonic, and hypotonic. Isotonic tonicity is where two solutions have an equal number of particles per volume of water. Hypertonic tonicity is a concentrated solution of water that will want to enter to disperse a solute concentration. Hypertonic tonicity is a concentration of solute water that will want to enter to disperse a solute concentration. Hypotonic tonicity, weakly concentrated solution of water will want to leave the concentration solute. Hypotonic tonicity, weakly concentrated solutions of water will want to leave the concentrate solutions. This is the lab that we did. Let's see if I can find anything. Diffusion. To exemplify the process of diffusion, you will need to place a solute of starch and glucose inside a bag of dialysis tubing. So the glucose diffuses and the starch doesn't because it's too big. Questions. What substance entered the bag? How do you know? Because it turned purple. So osmosis. So in diffusion, the glucose entered the dialysis tubing, but the starch didn't, and the starch didn't because it was too big. Diffusion, the glucose entered the dialysis tubing because it was small enough to go through, and the starch did not because it was too big. Osmosis. In this experience, you will collect the data of osmosis from distilled water into the test tube. 0.2 m, 0.4 m sucrose, 0.6 m sucrose, 0.8 m sucrose. Fill an 8-ounce cup 2/3 full, water asphyxiated. Obtain 5 strips of soaked dialysis tubing. Once the bags have been filled, tied off the end. So with osmosis, the final mass change. 8 ounce cups. 2.2 m bag matches the concentration of solution. 2 m bag set for 30 minutes. Osmosis, they went from less to more. If I remember that right. Yeah, it moves from a solution that is less concentrated to a solution that is more concentrated. So that's osmosis for you. All right, that was experiment 2. We got 3 to talk about. And that was reflex arcs and cutaneous receptors. Sensation 1 reflexes. Strike the ligaments. So in your reflexes arc, that's just kind of a reaction that you have. It's not developed by the brain. Your muscle spindles are shortening. So a reflex arc is just a natural reaction. It doesn't really go to the brain. It just reacts. Plantar reflex Babinski. Have a subject sit on a table bench, extend the length of the bench. Legs should be slightly rotated outward, applying firm but not painful pressure with the back of a reflex hammer. Draw a handle from the heel, foot, base. So Babinski's, if your neural tissue is working right, you will, your toes will curl and pull away. If you don't feel it, you'll push in. Babinski's sign, positive, push in, negative, pull away. Babinski's sign, positive, push in, negative, pull away. And that's just testing your nervous system. You're poking your foot. Positive reaction, you push in, negative, pull away. Sensation, mapping, temperature receptors, stamp and poke spots with a hot, cold. Okay. Sensation, procedure, ensure that equipped. So, sensation receptors. It's just you're mapping the sensation temperature receptors, pressing in different spots, because some spots have cold receptors, some spots have hot receptors. So those are separate. Sensation receptor mapping, hot and cold mapping. You have different receptors, hot and cold, and they're in different places. That's all that's doing the sensation hot and cold receptor mapping. It's just mapping out where the hot and cold receptors are. Touch receptors, 2-point threshold. Procedure, touch, obtain a single bristle, dorsal surface with the wrist, test grid. So you test on the grid, their head, and close their eyes, gently record the results on the grid. Next, obtain use of head to spin. 2-point discrimination, have a subject close their eyes and test each area listed data sheet. So with the touch receptors test with the point threshold, you're testing how many receptors are in that area. Some parts of your body have less touch receptors than other parts. The touch receptors you're testing different points on the body where you have different amounts of touch receptors in them. So some parts of your body will be more prone to being able to sense more bristles touching you than maybe other parts. If you were to use a brush, and that was that touch receptors, sensory one. I'll move on to oh my gosh, laboratory 3, laboratory 4, right here. Laboratory 4, visual activity. Snellen eye chart covering one eye with index. Attempt to read the line, the smallest letters. Repeat step 1 with the other eye. That's the visual activity. That's how you basically, it's just normal visual eye test to see how well you can see, what you can read. Visual activity is the regular eye test where you hold one eye and the other cover up the other eye and you read the lines, and that tells you how good your eyesight is. Astigmatism is where you stand 20 feet from the astigmatism chart, and it's like a wheel with lines, with parallel lines, and if you have astigmatism, you'll see some lines that are bolder, and if you don't have astigmatism, you'll see the clear parallel difference between the lines. So if you have astigmatism, do the astigmatism test where you will look at an eye chart and you'll see a spoke wheel of lines that are parallel to each other, and some will look more bolder, and that will tell if you have astigmatism. If you don't have astigmatism, it won't look bold. You'll be able to see the lines in between. Pupillary reflex. The amount of light allowed to project into the retina is regulated by a structure called a pupil. Pupillary reflex. So this is where you go in the dark room and you shine a light into a person's eye, and then when it constricts, when it sees the light, it's a reflex. A pupillary reflex test is when you shine a light into somebody's eye after they've been in the dark for so long that their pupil will constrict. That's the pupillary reflex. It's just a natural reflex that we have. After image blind spot test, you hold an index card with a color on it, and then when they use the color receptors, and you flip it over, and on the blank side they will see a different color on there, and that's because they're using up the after image blind spot test color receptors, or the color receptors in their eyes. So the after image blind spot test is where you hold a index card with a color on it for a minute, and they stare at it, and then when you flip it over, they'll see a different color on it. Say you're looking at red, you'll see blue on the white side, and that's because your color receptors have been used up on the other card, or on the other side of the card. After image blind spot test, you will hold an index card with a label on it for about a minute. Flip it over to the white side, and your partner will see a different color on that side because their color receptors have been used up. The blind spot test, you will hold a positive sign in a circle, in front. Close your left eye and place the plus sign directly in front of your right eye. So the plus sign goes in front of your right eye and the circle goes in front of your left eye. Then you move it forward in the blind spot test, and eventually they will lose the circle, and then you keep moving it forward and they'll see it again and it'll reappear. That's called the blind spot test. The blind spot test is where you hold a circle and a positive sign in front of somebody's eye, and you move forward while they stare at the positive sign, and they'll lose the circle eventually, and then it'll reappear once it gets closer. Sensation, taste, station, hearing. All right, the Rhines test. Striking a tuning fork on your shoe in order to begin the vibrations handle. When the subject describes a sound gone away, move the handle, extending down, tuning fork. Oh, so you hold on the Rhines test, you hold the tuning fork on the mastoid process, and then you hold it in front of the ear. That's called the Rhines test. The Rhines hearing test, you strike a tuning fork and hold it on the mastoid process, and then you move it over outside the ear, and they'll be able to hear it ringing. The Ronnie's hearing test, you strike a tuning fork and you hold it on the mastoid process until it disappears, then you hold it outside of the auditory canal until they can't hear it anymore. The Weber's test is where you strike a tuning fork and you hold it on top of the middle of their head till they can't hear it. Weber's hearing test, you strike a tuning fork on your shoe, and then you hold it on top of your partner's head until they can't hear it anymore. Weber's hearing test, you strike the tuning fork on your shoe and hold it on top of their head until they can't hear it anymore. Binaural localizing sound. Oh, that's where you like snap in different places and or make a sound, jingle a bell in different places, and your subject will close their eyes while you make a sound and move it around, and they try to pinpoint where the sound is coming from. Binaural localizing of sound, they try to pinpoint where the sound is coming from with their eyes closed. Binaural localizing sound, they try to pinpoint the sound where it's coming from with their eyes closed. Equilibrium. Oh, equilibrium is where you spin them around and see if they have good balance afterwards. Equilibrium is where you spin them around and see if they have good balance afterwards. So, Rhines test, you hold it on the mastoid process and then outside the auditory canal. See if they can hear it. And then, visual. And then the Weber's test, you hold it on top of their head and see if they can hear it there. All right, that's number 4. Oh snap. Reflexes, got that. So, do this one. Endocrine system, laboratory 5. Okay. With the endocrine system, hormones are like transmitters. Regulatory molecules secreted by a gland into the blood. Blood carries the hormone to target tissue. Hormones are like transmitters. Regulatory molecules secreted by a gland into the blood. Blood carries the hormones to a targeted tissue. Hormones are like transmitter. regulatory molecule secreted by a gland into the blood. Blood carries the hormones to targeted tissues. Hormone precursors. Prehormone and prohormone. There's prehormone and prohormone. Prehormone and prohormone. Prehormone is a precursor to prohormone. Prehormone is a precursor to prohormone. Prohormone is a long-chain chemical that is altered, cut, and spliced to make a hormone. Prohormone is a long-chain chemical that is altered, cut, and spliced to make a hormone. Prohormone is a long-chain chemical that is altered, cut, and spliced to make a hormone. Hormonal interactions. Synergism. Two hormones work together to produce the result. Synergism. Two hormones work together to produce the result. Synergism. Two hormones work together to produce a result. Synergism. Additive, complementary, permissive effects. Synergism. Additive, complementary, permissive effects. Synergism. Additive, complementary, permissive effects. Synergism. Additive, adding their overall effects together. Additive, adding all their overall effects together. Complementary. Each hormone stimulates a different step in the process. Complementary. Each hormone stimulates a different step in the process. Permissive effects. Hormone enhances the responsiveness of a target organ to a second hormone. Permissive effects. A hormone enhanced a responsiveness of a target organ to a second hormone. Antagonistic effect. Action of one hormone antagonizes the effect of another. Insulin and glucagon. Antagonistic effect. Action of one hormone antagonizes the effect of another. Insulin and glucagon. Antagonistic effect. Action of one hormone antagonizes the effect of another. Insulin and glucagon. Hormone action. The concentration of hormone in the blood reflects its rate of secretion. Accumulation is not typical. Target organs and liver. Half-life. Time required for plasma concentration to be reduced to 1/2 reference level. Half-life. Time required for plasma concentration to be reduced to 1/2 reference level. Half-life. Time required for plasma concentration to be reduced to 1/2 reference level. Only concentrations within normal physiological range produce normal tissue response. Only concentrations within normal physiological range produce normal tissue response. Only concentrations within normal physiological range produce a normal tissue response. Variations of hormone concentration within normal physiological range can affect the responsiveness of a targeted cell. Variations in hormonal concentrations within a normal physiological range can affect responsiveness of targeted cells. Priming effects. Upregulations. Priming effects. Upregulations. Priming effects. Upregulations. Desensitizing, downregulations. Desensitizing, downregulations. Desensitizing, downregulations. Priming effects. Increased number of receptors formed on targeted cells. Greater response by the targeted cells. Desensitization, downregulation. Decreased number of receptors on targeted cells equals less number of cell responses. Some hormones enter their target cell, bind to nuclear receptors proteins, cause the cell to begin replication, or result in the cell producing something. Some hormones enter their target cells, bind to nuclear receptor proteins, cause the cell to begin replication, or result in the cell producing something. Other hormones cannot enter the target cell, so bind to receptor proteins on the target cell membrane and activate second messenger system elicit response to the target cell. Other hormones cannot enter the target cell, So bind receptors proteins on the target cell membrane, activate a second messenger system, elicit a response in the target cell. Yet another regulatory molecule, autocrine and paracrine regulators do not travel through the blood. Autocrine and paracrine regulators do not travel through the blood. Autocrine and paracrine regulators do not travel through the blood. Autocrine, produced by a cell to affect itself, produce and act within the same tissue of an organ. Autocrine, produced by a cell to affect itself. Autocrine, produce and act within the same tissue of an organ. Autocrine, produce and act within the same tissue of an organ. Paracrine, produced within one tissue and regulates a different tissue. Paracrine, produced within one tissue and regulate a different tissue in the same organ. Paracrine, produced within one tissue and regulate a different tissue of the same organ. Prostaglandins, most diverse group of autocrine regulators. Prostaglandins is found in the immune system, reproductive system, digestive system, respiratory system, circulatory system, urinary system. Prostaglandins is found in the immune system, reproductive system, digestive system, respiratory system, circulatory system, urinary system. Prostaglandins, most diverse group of autocrine regulators. Prostaglandins is the most diverse group of autocrine regulators. Prostaglandins is the most diverse of autocrine regulators. Prostaglandins is found in the immune system, reproductive system, digestive system, respiratory system, circulatory system, urinary system. The pancreas is both an exocrine and an endocrine. Pancreas is both an exocrine and endocrine. Pancreas is both an exocrine and an endocrine. Pancreas is both an exocrine and an endocrine. Pancreas is both an exocrine and an endocrine. Secretions of insulin and glucagon. These are antagonistic hormones, which secretion in the insulin and glucagon. These are antagonistic hormones, which when insulin is released, it reduces the amount of glucagon. Secretion and insulin and glucagon are antagonistic hormones, where insulin is released, glucagon is reduced. Insulin, blood glucose. When insulin is released, blood glucose levels go down. When insulin is released, blood glucose levels go down. When insulin is released, blood glucose levels go down. Glucagon increases blood glucose levels, promotes glucogenesis with liver and skeletal muscle. Glucagon increases blood glucose levels, promote glycogenesis, liver and skeletal muscles. Glucagon increases blood glucose levels, promote glycogenesis, liver, skeletal muscles. Glucagon increases blood glucose levels, promote glycogenesis, liver, skeletal muscles. In diabetes, that is genetic pancreas disorders the body immune system. There's a lack of insulin-produced genetic problem. In diabetes, type 1 is believed to be a genetic problem. In type 1 diabetes is believed to be a genetic problem. Type 1 diabetes is believed a genetic problem. Type 2 diabetes, receptors for insulin on the body cells become desensitized. Chronically high levels of glucose blood. This can decrease the insulin production as well. In type 2 diabetes, receptors of insulin in the body cells become desensitized chronically to high levels of glucose in the body. This can lead to decreased insulin production as well. Diabetes, the receptors of insulin for the body cells become desensitized chronically to high levels of glucose in the body. This can decrease insulin production as well. In healthy people, as blood glucose levels rise, insulin is released and glucose is taken up by the cell's bodies and used for glucogen. Glucose levels rise, insulin is released, and glucose is taken up by the body cells. In order to, let's see, you are divided sample. Okay. Let's see. Blood sugar chart. Normal person without diabetes, 70 to 99 milligrams a deciliter. After 2 hours after a meal, as for a normal person, 140 milligrams deciliter for blood sugar chart. Blood sugar chart for somebody who has been fasting with diabetes is 80 to 130 milligrams per deciliter. And then after 2 meals after a meal, their blood sugar chart, person with diabetes should be reading around 180 milligrams per deciliter. A healthy person, their initial glucogen reading should be about 98. Glucose at 1 hour after eating, 196, and 126 after 2 hours. Diabetic person, their initial glucose reading should read around 127. Glucose after 1 hour of eating, 223. Glucose after 2 hours, 164. Initial glucose reading for a pre-diabetic patient should be 118 milligrams per deciliter blood glucose. After eating, 203. 1 hour after eating 203 milligrams per deciliter. And then glucose at 2 hours, 144 milligrams per deciliter. Okay. All right, now we are on muscles. My favorite. Muscle origin is where it originates from, like your bicep. It would originate on your oh my gosh, the top of your humerus, by your shoulder. So your bicep muscle would originate by your shoulder. Bicep muscle would originate by your shoulder. Insertion is the distal end of that muscle. It would be the distal end of the bicep. It'd be the insertion point in the elbow. Insertion would be the distal end of the muscle. The insertion would be the distal end of the muscle. Origin would be the closer end of the muscle. Structure of skeletal muscle. Fascicles, bundle of muscle cells separated by paramycium, muscle cells separated from each other by endomycium. Fascicles, bundle of muscle cells separated by paramycium, muscle cells separated from each other by endomycium. Fascicle, bundle are bundles of muscle cells separated by paramycium, muscle cells separated from each other by endomycium. Fascicles are bundles of muscle cells. Fascicles are bundles of muscle cells. Fascicles are bundles of muscle cells. The sarcolemma is the cell membrane for the muscle cell. Sarcolemma is the cell membrane for the muscle cell. Sarcolemma is the cell membrane for the muscle cell. Sarcolemma is the cell membrane for the muscle cell. Sarcoplasm, muscle cell. Cytoplasm, sarcoplasm, muscle cell, cytoplasm, sarcoplasm, muscle cell, cytoplasm. Sarcoplasmic reticulum stores calcium when muscle is at rest. Sarcoplasmic reticulum stores calcium when muscle is at rest. Sarcoplasmic reticulum stores calcium when muscle is at rest. Sarcoplasmic reticulum stores calcium when muscle is at rest. T-tubules conduct action potential through muscle cell. T-tubules conduct action potential through muscle cell. T-tubules conduct the action potential through muscle cell. T-tubules conduct action potential through muscle cell. Motor unit, all muscle fibers are innervates. Each somatic neuron together with all muscle fibers, it innervates. Motor unit. Each somatic neuron together with all muscle fiber, it innervates. Each muscle fiber receives a single axon terminal from a somatic neuron. Each muscle fiber receives a single axon terminal from a somatic neuron. Each muscle fiber receives a single axon terminal from a somatic neuron. Each muscle fiber receives a single axon terminal from a somatic neuron. Each muscle fiber receives a single axon terminal from a somatic neuron. Each axon can have many terminal branches to innervate an equal number of fibers. Each axon can have many terminal branches to innervate an equal number of fibers. Each axon can have many terminal branches to innervate an equal number of fibers. Each axon can have many terminal branches to innervate an equal number of fibers. When somatic neuron is activated, all muscle fibers in its motor unit contract. When a somatic neuron is activated, all muscle fibers in its motor unit contract. When a somatic neuron is activated, all muscle fibers in its motor unit contract. Innervation ratio, 10 to 10 to 1 to 10. More neurons per group of muscle fibers equals greater control, precision. Innervation ratio, 10 to 10, 1 to 10, to 1 to 10. More neurons per group of muscle fibers equals greater control, precision. Innervation ratio, ratio of 10 to 10 to 1 to 10. More neurons per group of muscle fibers equals greater control, precision. Recruitment, larger and larger motor units are activated to produce greater strength. Recruitment, larger and larger motor units are activated to produce greater strength. Recruitment, larger and larger motor units are activated to produce strength. Neuromuscular junction, the union of nerves and muscle. 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 Motor end plate on the muscle cell membrane. 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 Mechanisms of contraction. Each myofibril contains myofilaments. Thick filaments equals myosin. Thick filaments equals myosin. Thick filaments equals myosin. Myosin contains mostly thick filaments. Thin filaments, actin. Thin filaments, actin. Thin filaments, actin. Thin filaments, actin. Actin contains a thin filament. A band contains both thick and thin filaments. Center of each I band is myosin M-line. Center of each I band is myosin M-line. Center of each I band is myosin M-line. Mechanisms of contraction. Each myofibril contains myofilaments. Thick myofilaments equals myosin. Myosin contains mostly thick filaments. Thin filaments equals actin. Actin contains thin filaments. A band contains both thick and thin filaments. Center of each I band is myosin. A band contains both thick and thin filaments. A band contains both thick and thin filaments. Sarcomere contraction. A bands move closer together, do not shorten. A bands move closer together, do not shorten. A bands move closer together, do not shorten. I bands decrease in length, occurs because of sliding thin filaments over and between thick filaments. I-band decrease in length occurs because of sliding of thin filaments over and between thick filaments. I-band decrease in length occurs because of sliding of thick filaments over between thick filaments. I-band decrease in length occurs because of sliding of thin filaments over between thick filaments. I-band. Mechanisms of contraction. So voltage gated comes before chemically gated. Voltage gated comes before chemical gated. Voltage gated comes before chemical gated. Voltage gated, calcium. Chemically gated, sodium, potassium. Chemically gated, sodium, potassium. Chemically gated, sodium, potassium. Chemically gated, sodium, potassium. Voltage gated, calcium. Voltage gated, calcium. Mechanisms of contraction. AP travels down the motor neuron to the terminal bouton. Voltage-gated calcium channels open. Calcium diffuses into the bouton. Calcium binds to vesicles, neurotransmitter. Acetylcholine released into the neuromuscular cleft, neuromuscular junction. Acetylcholine binds to receptors on motor end plate of the muscle fiber. Chemically gated channels for sodium and potassium open. Positive ions depolarizing travel down T tubules. Sarcoplasmic reticulum releases stored calcium. Calcium binds to troponin on actin filaments. Troponin, tropomyosin complex moves active binding sites for myosin on actin undisclosed. Mechanisms of contraction. Voltage gated, calcium. Chemically gated comes after sodium, potassium. Mechanisms of contraction. AP travels down motor neuron to terminal bouton. Voltage gated calcium channels open. Calcium diffuses into bouton. Calcium binds to vesicles, neurotransmitter. Acetylcholine released into neuromuscular cleft, neuromuscular junction. Acetylcholine binds onto receptors on motor end plate of the muscle fiber. Chemically gated channels for sodium and potassium open. Positive depolarization travels down T tubules. Sarcoplasmic reticulum releases calcium. Calcium binds to troponin and actin filaments. Tropinamycin complex moves active binding site for myosin on actin is disclosed. Regulations of contraction. Regulation of cross-bridge attachment is actin due to tropomyosin and troponin regulatory proteins. Regulation of contraction. Regulation of cross-bridge attachment to actin due to tropomyosin and troponin regulatory proteins. Regulatory contraction. Regulatory cross-bridge attachments actin due to tropomyosin and troponin regulatory proteins. Tropomyosin, troponin regulatory proteins. Myosin's turn. Think of myosin as small ratchets. Each ratchet head has a binding site for ATP. When the actin site is exposed, the myosin heads use ATP to attach themselves to the actin. ADP and P are still on the head. When the head binds to actin, the P is kicked off and the head changes position. Bends power, stroke. A new ATP then comes over to the myosin head and attaches to it. The myosin head is released from actin and goes back to its resting orientation. Troponin, tropomyosin recover the actin building site. Calcium is pumped back into the sarcoplasmic reticulum. ATP is needed there too. Myosin's turn. Think of myosin as small ratchets. Each ratchet head has a binding site for ATP. When the actin site is exposed, the myosin head uses ATP to attach themselves to actin. ADP and P are still on the head. When the head binds to actin, the P is kicked off and the head changes position, bends, power stroke. A new ATP comes from over the myosin head and attaches to it. The myosin head is released from the actin and goes back to its resting orientation. Troponin and tropomyosin recover the actin binding site. Calcium is pumped back into the sarcoplasmic reticulum. ATP is needed there too. Myosin's turn. Think of myosin as small ratchets. Each ratchet head has a binding site for ATP. When the actin site is exposed, myosin heads use ATP to attach themselves to actin. ADP and P are still on the head. When the head binds to actin, the P is kicked off and the head changes position, bends, equal power stroke. A new ATP then comes over to the myosin head and attaches to it. The myosin head is released from the actin and goes back to its resting orientation. Troponin and tropomyosin recover the actin binding site. Calcium is pumped back into the sarcoplasmic reticulum. ATP is needed there too. And I believe that is all. Hope you all enjoyed. Comment, like, subscribe.